In the last chapter, we looked at the discovery of DNA and the structure of DNA and RNA. And in this chapter, we are going to focus on the functions of DNA and RNA. So we'll take a look at DNA replication and protein synthesis. So we learned that DNA is the genetic material and it is passed from parent to offspring. And because of this, DNA, when it's replicated, must be done with extreme accuracy because any mistakes that are made then can be passed on. Those are called mutations, and they can be passed on and basically become a permanent part of the genome. The second thing is that DNA is used as the instructions to create proteins. And so the gene, as we saw that, on the DNA, we have the gene and we have the non-coding portions of that. So the gene is going to be read and then transcribed, okay, into mRNA. And then it's going to be translated to actually produce the protein. And this process of transcription and translation is referred to as gene expression. Okay, so gene expression. The flow of this information, so the flow of information from DNA into RNA, messenger RNA, and into protein is called the central dogma. Okay. And basically what this is it's the mechanism of that one gene, one protein hypothesis that we learned about from BIOL and Tatum. And so we're going to have replication that takes place, the DNA replication that takes place. We have the transcription where we go from DNA to messenger RNA, and then translation where we actually translate that mRNA into the protein. So in order for DNA to be passed down from parent to offspring, it has to be replicated. We have to provide an exact copy of the DNA. And when we got that structure of DNA provided by Watson and Crick, it kind of gave us maybe a little information as far as how DNA is copied. But when we separate the strands of the double helix, it provides essentially two templates. We can build off of either template, but we weren't 100% sure exactly how that happened. Now we know that DNA replicates by a method called semi-conservative replication. And each strand is going to serve as a template. And so that's what you see here in this figure, that DNA replicates by this semi-conservative. So the parent DNA, again, remember that DNA is double-stranded. So the DNA will separate, and we've got the parent DNA here in blue. And so each one of those would be used as a template strand. And we would have a new DNA, or the daughter strand, that is created. And essentially, in this case, in these cells, each cell is going to receive a copy of the parent DNA. And so this is the method by which it happens. The other methods that were proposed were this conservative or this dispersive manner, and we realized that that's not how it worked. And this was done in an experiment by Messelson and Stahl, and they're the ones that essentially figured all of this out. There are a couple of different processes when we look at DNA replication in bacteria versus DNA replication in eukaryotic or more complex cells. DNA replication in bacteria has been studied extensively because bacteria have a small genome. And so if you look at, as an example, E. coli, it has 4.6 million base pairs. Okay, and so when we talk about a base pair, we're talking about, adenine, thymine, or guanine, cytosine. So E. coli has 4.6 million base pairs. And remember, prokaryotes only have one circular chromosome. And this takes about 42 minutes. So it doesn't take very long for this process to occur, so it makes it readily available for studying. Now, the process of DNA replication can get very, very complicated. And I'm going to basically just give you the very tip of the iceberg when it comes to DNA replication. I'm not going to go through every single enzyme and every single step of the process. We're just going to take a basic overview of how it works. But just keep in mind, just like when we talked about, cellular respiration, that it requires an immense number of enzymes and it requires a lot of moving parts for DNA replication to occur this rapidly and for it to occur without errors or with very few errors. So when we look at DNA replication in bacteria, we're going to have a main enzyme that's called DNA polymerase. And there are three main types of DNA polymerase that we have. And when we build DNA, when we're replicating DNA, it does require energy. It does, and that's in the form of ATP, essentially. And it's going to be present in these phosphate bonds that are identical to ATP. DNA replication begins at a specific sequence of the nucleotides, so a specific sequence of the base pairs called the origin of replication. And that's kind of how the DNA knows, like, this is the place to replicate. So we have to relax, because DNA is supercoiled. It's bound up so that it can fit in that chromosome. So it has to be uncoiled, and then it has to be essentially unzipped. We have to unzip that helix in order to free the base pairs so that we can bring in complementary nucleotides. And as the DNA is separated, this is done by an enzyme called helicase. It breaks those hydrogen bonds. And as the DNA opens up, we get this area called a replication fork. And this replication fork is essentially that split or that gap where the two halves or the two pieces of the helix have separated. And we have two replication forks that are formed so that we can get what we call a bidirectional replication. So remember when we were looking at the structure of DNA, we had that 5' end and the 3' end. And I told you that DNA, when it was put together in its double helix, is anti-parallel, meaning that, we run on one side, we run in a 5' to 3' direction, and on the other side, we run in a 3' to 5' direction. So we need two replication forks so that we can build the new DNA in both directions at the same time. And then we have a primer that's going to be built. This is a helper of RNA that comes in and helps us kind of get the deal started. And so you've got this image here in your textbook, and there's a lot of stuff, kind of going on here. But basically what we've got, here's our parent DNA. Okay, so our parent DNA is going to be in the blue. And again, we've got that 5' end and the 3' end. And so we're building, towards the 3' on this side and towards the 5' on this side. And So this is our template. This is called the leading strand. And in this strand, as we're building in the 5' to 3', because it's going to be complementary, right? It's anti-parallel. So as we're building this leading strand here, we can make it continuous. It can be brought in, and we're just going to bring in nucleotide after nucleotide, and we have this thing called a sliding clamp, basically, that's leading us in that direction. Okay, and the helicase is just going to keep unzipping as we move through here. But on the other side, okay, this is called the lagging strand or the discontinuous strand. We can't build an entire unit continuously like we do in this way. When we're building from a 5' to a 3', we have to bring in little pieces at a time, and these are called Okazaki fragments. So we're going to build a little piece at a time, and then that's going to essentially be glued together with an enzyme called DNA ligase. So it's a little bit different process. So on our leading strand, it's just continuous. But on our lagging strand, we add just a little piece at a time. And we're only going to unzip one little area at a time. And when this gets all back together, then this basically will kind of come a little bit back together. Not all the way, because we've got, 2 strands here. But the whole thing is not going to be unzipped at once. We're only going to do a piece at a time. Now, after the complete chromosome has been replicated, so we've gone down the entire length of the chromosome and we've replicated it, we have the termination process, and we don't really know a lot about that. We know that the DNA they're basically kind of locked together. So the parent strand and now our new strand that we've created is kind of locked together. So they have to be interlocked and they have to be separated, and there's some enzymes that do that. But we don't really know exactly how that termination sequence begins or how that happens. We just know that there are some enzymes that are involved in that process. When we look at eukaryotic DNA replication, the processes are pretty similar, and a lot of them are, in fact, the same. The replication process occurs much more slowly in eukaryotes, only about 100 nucleotides per second versus about 1,000 nucleotides per second in prokaryotes. One of the differences is that eukaryotes have linear chromosomes, and what will happen is once the DNA helicase comes in and unzips it, instead of unzipping it really at one place, it's going to unzip it at multiple points along the linear chromosome, and we'll get building of that new strand out from there, and eventually these areas here will meet. We have similar enzymes. The structure is a little bit different, but we still have helicase, and we have gyrase and DNA polymerase. So a lot of the enzymes are the same. The really big difference that we see between prokaryotic and eukaryotic DNA replication is with these areas called telomeres. The ends of all linear chromosomes have these little areas of non-coding sequences, and those are called telomeres. And basically, they prevent the coding sequences from being lost. You see there in humans, we have a 6 base pair sequence, TTAGGG, that's repeated 100 to 1,000 times to form this telomere. And during this process, we have to use a specific enzyme called telomerase that is going to basically complement this overhang and prevent this little piece that's been formed of the new DNA from being lost. So that's really the huge major difference between DNA replication in prokaryotes and eukaryotes is that we have the addition of these telomeres at the ends of the chromosomes, and so we have a special enzyme called telomerase that is needed for those.